There is a man in a rehabilitation center in Baltimore who has not recognized his wife's face in 11 years. His name in the research literature is Eugene Pauly, and the damage to his medial temporal lobe, caused by viral encephalitis in the early 1990s, was so severe that it abolished nearly every trace of his declarative memory. He cannot form new long-term memories. He cannot tell you what he had for breakfast or whether he has had breakfast at all. He cannot describe the house he has lived in for decades, or draw a map of his neighborhood, or tell you where the kitchen is when asked directly. And yet, each morning, Eugene rises from bed, walks down the hall, opens the correct cabinet, selects the appropriate bowl, pours cereal and milk in the right sequence, and returns to his chair without confusion, without error, and without any conscious awareness that he is doing any of it. When researchers from the University of California, San Diego studied him in the mid-1990s, they were confronted by a phenomenon that seemed, on the surface, to be paradoxical: a man with no functional memory performing complex, contextualized, correctly sequenced behaviors with remarkable reliability. What they were actually observing was not a paradox. It was one of the clearest demonstrations in the history of neuroscience of where habit lives in the human brain, and why it is categorically different from everything we typically mean when we talk about knowledge, intention, or choice. The story of habit has been told many times, but it has rarely been told precisely. It has been domesticated into a self-help narrative, compressed into a productivity formula, and presented as a set of techniques that, if applied with sufficient discipline, will rearrange the trajectory of a life. The science behind it is considerably stranger, considerably more humbling, and considerably more interesting than that narrative suggests. It begins not with motivation, not with willpower, not with the crystallizing decision to change, but with a structure buried so deep in the architecture of the brain that it predates the cortex, the seat of conscious thought, by hundreds of millions of years. To understand what habits actually are, and why even the smallest of them carry such disproportionate influence, it is necessary to begin with a piece of anatomy that most people have never considered, and whose function contradicts almost every popular assumption about how human behavior is generated. The basal ganglia are a collection of interconnected nuclei situated deep within the cerebral hemispheres, surrounding the thalamus and lying beneath the cerebral cortex. They are evolutionarily ancient, present in forms recognizable across all vertebrate species, in fish, in reptiles, in birds, in mammals, which suggests that whatever function they serve was important enough to be conserved across an immense span of biological time. In humans, they were long understood primarily as motor control structures, their relevance acknowledged mainly in the context of the diseases that destroy them: Parkinson's disease, Huntington's disease, conditions in which movement becomes rigid, tremulous, or uncontrolled. But beginning in the 1990s, a series of experiments conducted by neuroscientist Anne Graybiel and her colleagues at the Massachusetts Institute of Technology began to reveal a function of the basal ganglia far more fundamental than motor coordination alone. Graybiel's laboratory implanted recording electrodes in the striatum, the largest component of the basal ganglia, of rats learning to navigate a T-shaped maze. A click would sound, a gate would open, and the rat would move through the maze to find a piece of chocolate at one of the ends. What happened inside the striatum across the weeks of training was not what anyone had predicted. Initially, the neurons fired almost continuously throughout the entire maze run, flickering with activity at each moment of decision and movement. But as the behavior became practiced, as the route from the gate to the chocolate became habitual, the pattern of neural activity changed dramatically. The constant mid-route firing diminished. Instead, two distinct bursts of activity appeared, one at the very start of the run, triggered by the click and the opening gate, and another at the very end, when the chocolate was reached. The neurons in between fell almost entirely silent. What Graybiel had observed was the brain performing what she would call chunking, the compression of an entire behavioral sequence into a single, unified neural unit. Where previously the brain had processed each individual step of the maze, run as a discrete decision requiring attention and evaluation. It had reorganized the whole sequence into an efficiently encoded package: one trigger, one integrated routine, one reward signal. The cognitive cost of performing the behavior had been reduced, in neural terms almost to zero. The brain no longer needed to consciously monitor or direct the sequence. It needed only to initiate it. This is what a habit is at the level of neuroscience, not a virtue, not a discipline, not an achievement of willpower. It is a neural compression, an economy of processing that allows the brain to perform frequently repeated behaviors with minimal expenditure of attentional resources, freeing those resources for tasks that are genuinely novel and uncertain. The brain is, in this sense, a ruthlessly efficient system that constantly seeks to convert deliberate action into automatic execution. Every time you brush your teeth, navigate a familiar route home, or pour your morning coffee in a precise, unvarying sequence without consciously thinking about any step of it, you are observing the basal ganglia performing exactly what Graybeel's rats demonstrated in that T-shaped maze. The behavior has been chunked. Its execution has been transferred from the slow, deliberate, metabolically expensive machinery of the prefrontal cortex to a structure that operates faster, more economically, and almost entirely beyond conscious awareness. The implications of this transfer are immense, and they cut in multiple directions simultaneously. On one hand, habituation is the mechanism that makes complex human lives possible. A skilled surgeon who has performed the same procedure hundreds of times operates partially through habit. The fine motor sequences encoded so deeply in neural architecture that they can be executed with a precision that deliberate conscious control could not match. A concert pianist navigating a demanding passage of Rachmaninoff does not consciously direct each finger. The fingers, through years of practice, have been delegated the task by a brain that recognized the efficiency of habitual execution. The cognitive load of living would be crushing if every action demanded the full attentional resources of the prefrontal cortex. Habituation is how we function at all in a world requiring thousands of coordinated actions, per Dalton Y. On the other hand, the very efficiency of this system, its capacity to operate below the threshold of conscious awareness, means that habits once formed are extraordinarily resistant to deliberate revision. This is the feature that makes them so powerful as behavior patterns and so frustrating as obstacles to change. The basal ganglia, unlike the prefrontal cortex, do not maintain a running evaluation of whether a behavior is currently desirable. They do not update their programming in response to consciously held values or intentions. A habit encoded in the striatum will be triggered by its associated cue, regardless of whether the person performing it has decided, thoughtfully and sincerely, that they no longer wish to perform it. The hunger for cigarettes activated by the smell of coffee, the route toward the fast food restaurant, taken almost involuntarily on the commute home, the sequence of evening behaviors that ends in hours of passive screen consumption, despite a sincere intention to sleep earlier. These are not failures of character or insufficient resolve. They are chunked behavioral sequences, executing precisely as the basal ganglia were designed to execute them, in response to the cues that originally trained them into existence. Understanding this distinction between the formation of a habit and the conscious desire to have one is where the science diverges most sharply from the self-help narrative that has been constructed around it. Popular accounts of habit formation tend to emphasize motivation, wanting the outcome badly enough, connecting behavior to a compelling future vision, harnessing the emotional drive toward a better life. These are not without relevance, but they address the prefrontal cortex, the deliberate system, the part of the brain that plans, evaluates, and chooses, while habits are created and maintained by a system that largely ignores what the prefrontal cortex has decided. The question is not whether a person wants to change. The question is how the cue routine reward loop of the basal ganglia comes to encode a new behavioral sequence in the first place, and why size. The scope and ambition of the initial behavior matters so much in that encoding process. In 2010, health psychology researcher Philippa Lally and her colleagues at University College London published a study that attempted to measure, with actual empirical data, how long it takes for a new behavior to become automatic, to transition from deliberate action requiring conscious intent to the kind of routine execution that characterizes a genuine habit. The study tracked 96 people over 12 weeks as they attempted to form a new daily habit. Behaviors as simple as drinking a glass of water with lunch, eating a piece of fruit in the afternoon, or taking a short walk after dinner. Participants rated the automaticity of their target behavior each day, using a validated scale that measured how little conscious attention the behavior required to execute. The findings defied the popular 21-day, rule that had circulated since Maxwell Maltz's 1960 self-help book Psycho, Cybernetics, and confirmed only that the popular rule was essentially fabricated. The actual average time to reach a plateau of automaticity was 66 days, and within that average lay a range of considerable variation, from as few as 18 days for very simple behaviors to as many as 254 days for more complex ones. What the study also revealed, and what received far less popular attention, was a finding about missed days. The researchers discovered that a single missed repetition of the target behavior did not measurably set back the formation of the habit. The asymmetry between consistency and occasional disruption matters enormously for how we understand the actual mechanism of habit formation. The brain's chunking process is not fragile. It does not reset on the occasion of a missed cue, routine, reward cycle. What it requires is not perfection, but sufficient repetition within a consistent context. And this is where the size of the initial behavior becomes critical in a way that is genuinely non-intuitive. The conventional approach to behavioral change is goal-driven. A person identifies an outcome they want: better physical fitness, a regular reading practice, a more disciplined approach to work, and designs a behavior commensurate with the scale of that ambition. Someone who wants to become physically fit resolves to exercise for 45 minutes each morning. Someone who wants to read more commits to a chapter per night. The logic is straightforward. Large goals require substantial effort, and substantial effort demonstrates the seriousness of the commitment. What this approach misunderstands is the neuroscience of habit formation. Large initial behaviors carry with them large initial motivational requirements. They depend on the prefrontal cortex, on deliberate intention, on the conscious marshaling of attention and effort, in exactly the way that established habits do not. And crucially, the prefrontal cortex is the most metabolically demanding component of the brain, the system most vulnerable to fatigue, stress, competing demands, and the accumulated cognitive load of an ordinary day. Psychologist Roy Baumeister proposed in a landmark 1998 paper that self-control functions like a muscle, that the capacity for effortful regulation depletes with use, leaving a person less capable of sustained willpower as demands accumulate across a day. He termed this ego depletion. The original experiments were elegant. Participants required to resist eating chocolates in favor of radishes subsequently gave up faster on an unsolvable puzzle than participants who had faced no prior self-control demand. The theory became enormously influential, shaping dietary advice, productivity frameworks, and arguments for the structural importance of morning routines. What happened, subsequently, was considerably more complicated. A large-scale multi-laboratory replication attempt in 2016, involving 23 independent laboratories and more than 2,000 participants, failed to reproduce the ego depletion effect at a statistically meaningful level. The confidence interval for the effect size straddled 0. This did not definitively disprove the concept. Subsequent research has suggested the original model was oversimplified, that self-control may involve motivational factors rather than a single depletable resource, and that beliefs about willpower may themselves influence its apparent availability. But the replication failure injected significant uncertainty into the strongest version of the claim. What has emerged from this contested evidence is a more nuanced picture. Willpower is not simply a limited fuel tank that empties with use, but it is also not a boundless resource. And deliberate effort, dependent self-regulation, is less reliable as a behavior change mechanism than the self-help industry had built its entire architecture around. Psychologist Roy Baumeister proposed in a landmark 1998 paper that self-control functions like a muscle, that the capacity for effortful regulation depletes with use, leaving a person less capable of sustained willpower as demands accumulate across a day. He termed this ego depletion. The original experiments were elegant. Participants required to resist eating chocolates in favor of radishes subsequently gave up faster on an unsolvable puzzle than participants who had faced no prior self-control demand. The theory became enormously influential, shaping dietary advice, productivity frameworks, and arguments for the structural importance of morning routines. What happened subsequently was considerably more complicated. A large-scale multi-laboratory replication attempt in 2016, involving 23 independent laboratories and more than 2,000 participants, failed to reproduce the ego depletion effect at a statistically meaningful level. The confidence interval for the effect size straddled zero. This did not definitively disprove the concept. Subsequent research has suggested the original model was oversimplified, that self-control may involve motivational factors rather than a single depletable resource, and that beliefs about willpower may themselves influence its apparent availability. But the replication failure injected significant uncertainty into the strongest version of the claim. What has emerged from this contested evidence is a more nuanced picture. Willpower is not simply a limited fuel tank that empties with use, but it is also not a boundless resource. And deliberate effort, dependent self-regulation, is less reliable as a behavior change mechanism than the self-help industry had built its entire architecture around. This is precisely where small behaviors, sometimes dismissively called tiny habits, acquire genuine scientific justification. The argument for beginning with very small behaviors is not a concession to laziness or a hedge against ambition. It is a structural insight about how the basal ganglia actually encode routines. B.J. Fogg, a behavior scientist at Stanford University's Persuasion Technology Lab, spent years developing and studying a framework built around the smallest possible versions of target behaviors. Not a morning exercise routine, but a single push-up immediately after getting out of bed. Not a journaling practice, but a single sentence written each evening. Not a meditation discipline, but 3 deliberate breaths taken after brewing coffee. The principle behind these reductions is neurological rather than motivational. A behavior small enough to require essentially no volitional effort, no activation of the effortful, deliberate prefrontal system, can be initiated and completed consistently enough in a consistent enough contextual setting to trigger the chunking process in the basal ganglia before willpower ever becomes relevant. The consistent contextual setting is, it turns out, at least as important as the behavior itself. Wendy Wood, a behavioral psychologist at the University of Southern California, whose research on habit spans more than 3 decades, has shown with considerable empirical rigor that habits are fundamentally tied to context, that the cue triggering and habitual behavior is almost always a feature of the physical or social environment in which the behavior was originally performed. Wood's research demonstrated that when people's environments change, when they move to a new city, change jobs, or experience a major life disruption, old habits become unexpectedly weak, and new ones form with unusual ease. The brain's chunking mechanism is not merely storing the behavioral sequence. It is storing the association between a specific environmental configuration and the initiation of that sequence. Remove or alter the environmental cue, and the habit loses its trigger. Install a new environmental configuration as a consistent context for a new behavior, and the encoding of that behavior into automatic execution proceeds far more readily than any amount of deliberate motivational effort could produce. This has led Wood to a conclusion that runs directly counter to the dominant narrative of individual willpower as the engine of behavioral change. The most effective interventions for sustained behavior change, her research suggests, are not those that deepen a person's motivation or strengthen their resolve, but those that restructure the environment in which the person operates. A person who wants to eat more fruit will be more reliably successful if they place fruit on the kitchen counter at eye level than if they commit, however sincerely, to choosing fruit over less healthy options. A person who wants to exercise regularly will be more reliably successful if they sleep in their workout clothes or drive a route to work that passes the gym than if they maintain a vivid mental image of the life they want. The environment does not merely support the habit. For a mature habit, the environment essentially is the cue, and the cue essentially is the behavior. Restructuring context restructures behavior at a level below conscious choice. There is something unsettling in this conclusion for those who prefer to understand themselves as rational agents whose behavior flows from consciously held values and intentions. Research suggests that approximately 40 to 45% of human daily behavior consists of habitual actions. Behaviors performed in the same location at the same time with the same outcome each day, requiring essentially no deliberate decision. Nearly half of what we do, each day, we do not choose, we execute. The decision was made once, long ago, by the conditions under which the basal ganglia first encoded the sequence. What presents itself as ongoing choice is, in nearly half of all instances, the triggering of an already written program. This figure changes the ethical and practical stakes of habit formation significantly. If the person you become is substantially shaped by behavioral patterns, encoded early and executed automatically thereafter, then the moments of genuine choice, the moments when the prefrontal cortex is actually directing behavior, rather than merely observing what the basal ganglia have already initiated, are precious and relatively rare. And the choices that matter most may not be the large, dramatic ones, the decision to quit or to begin, the resolution made at a moment of crisis or inspiration. The choices that may carry the greatest long-term consequence are the small, contextual, repetitive ones, the ones that, through sufficient repetition in sufficient context, eventually disappear from the domain of choice entirely and settle into the quiet, continuous automaticity of who we simply are. This is where an important and underappreciated dimension of habit research enters the picture, one that shifts the analysis from the mechanical to the deeply personal. Research published in 2025 in the journal Applied Psychology, Health and Well-Being documented a significant positive correlation between habit strength and identity, finding an effect size large enough to suggest that what we do reliably and what we consider ourselves to be are not merely correlated but mutually constitutive. The habit shapes the identity, and the identity subsequently reinforces the habit. A person who has performed a behavior consistently enough to feel that it reflects who they are does not experience the behavior as an effortful choice or a discipline. They experience it as an expression of self. And the psychological resistance to abandoning a behavior that reflects who one is considerably more robust than the resistance produced by any consciously held commitment. This bidirectional relationship between habit and identity illuminates why the sequence in which behavioral change is attempted may matter as much as the behavior itself. The conventional approach, pursue the behavior, hope the identity follows, is neurologically backward. A person who exercises reluctantly 3 times per week, fighting the habit each time, drawing on willpower reserves that may or may not hold, is attempting to build an identity from the top down, using the effortful prefrontal system to generate behaviors that have not yet been transferred to the automatic execution of the basal ganglia. A person who begins with behaviors small enough to be performed without effort, repeated consistently enough to become automatic, until the pattern of repetition produces a genuine change in self-perception. This person is working with the architecture of the brain rather than against it. The identity does not precede the habit. It is the residue of the habit's accumulation. The accumulation itself, the mathematics of small, consistent behaviors compounding across time, deserves more careful attention than it typically receives. Behavioral economists and psychologists who study long-term patterns of change have noted that human beings are particularly poor intuitive calculators of exponential processes. We understand linear accumulation reasonably well. $10 added to a savings account 10 times yields $100. We understand immediate, dramatic change, a single event that transforms a situation overnight. What we systematically underestimate is the effect of small, consistent inputs compounded across time, the kind of growth curve that begins imperceptibly and then accelerates into outcomes that seem, in retrospect, to have arrived suddenly, though they were in fact the mathematical inevitability of a sustained practice. A person who reads 10 pages of a serious book each evening, 10 pages, roughly 15 minutes of focused engagement, will, at the end of a year, have read approximately 12 to 15 books of substance. At the end of a decade, over 120. The behavior does not feel significant on any given evening. The compounding of it, across years, is transformative in a way that a single intensive month of reading could never replicate. The same arithmetic applies to physical conditioning, to language acquisition, to financial accumulation, to the development of any complex skill. The brain, through the chunking mechanism of the basal ganglia, is precisely suited to this kind of accumulation, because it makes the consistent repetition progressively cheaper, in cognitive terms, as the behavior becomes more automatic. What happens at the intersection of this compounding in the context? Dependency of habits is perhaps the most practically significant finding in all of this research. Grabiel's group at MIT identified what they called keystone habits, behavioral routines, that, once established, appear to trigger the spontaneous formation of other positive routines through a mechanism that researchers have not fully characterized, but that appears to involve both the changed self-perception that accompanies behavioral change and the restructuring of daily routines that any new consistent behavior requires. Regular exercise, in multiple large-scale longitudinal studies, predicts improved dietary choices, reduced impulsive spending, better sleep discipline, and higher reported productivity, outcomes that have no obvious mechanistic connection to physical movement, but that seem to arise from the identity shift and routine restructuring that a consistent exercise habit produces. The small behavior does not operate in isolation. It operates as a structural intervention in the ecology of a person's daily life, altering the context in which dozens of other behavioral choices are made, and frequently cascading into changes that were never directly targeted. This cascade effect is what the science of small habits ultimately reveals about the relationship between action and identity, between what we do and who we become. The brain is not neutral territory, passively registering the decisions a conscious self makes about how to live. It is an active, self-organizing system, continuously encoding the patterns of behavior it observes, continuously compressing the frequently repeated into the automatically executed, continuously updating the neural architecture of who a person is based on what that person consistently does. The self we experience as a stable, conscious agent choosing freely among options is, at least in substantial part, the accumulated output of behavioral patterns we chose. In a more limited sense, at some earlier point, in which the basal ganglia have since taken custody of. There is something genuinely liberating in this understanding. Once the initial discomfort of reduced agency passes, it means that the architecture of a life is not primarily determined by large dramatic acts of will performed in moments of inspiration. It is determined by the small, contextually consistent, repeatedly performed behaviors that the brain quietly elevates from choice to character. The implications extend well beyond the domain of individual self-improvement. They touch the design of schools, of workplaces, of healthcare environments, of public spaces, any context in which human behavior matters, and in which the conditions shaping that behavior can be deliberately arranged. If nearly half of all daily behavior is habitual, then the environments we inhabit are not backdrops to our choices. They are co-authors of our behavior, shaping it through the contextual cues that trigger behavioral sequences long after the conscious decision that initiated the sequence has been forgotten. Eugene Polly, the man without functional memory who can still navigate his kitchen and return to his chair with his cereal, did not choose those behaviors each morning. He did not remember that he had performed them before. He did not draw on motivation, or willpower, or a vision of the self he wanted to become. What he accessed, in the absence of every other cognitive capacity, was the one system that the virus had not reached, the basal ganglia, deep and ancient and efficient, holding in their encoded circuitry the patterns of a life that his conscious mind could no longer recall. The habits remained when almost nothing else did, which suggests, perhaps more clearly than any experiment conducted under controlled conditions, what habits actually are, not the supplements to a life lived deliberately, but the substrate of it, the accumulated, compressed, automatically executed residue of everything a person has chosen to do, quietly and consistently, over the course of a lifetime.